अब हम बात करेंगे एंटीबॉडी हाइब्रिडाइजेशन की प्रोटीन्स के साथ विच आर immobilized on the nylon membrane. Again, this is very important to wear gloves throughout this procedure so as not to contaminate any protein from the hands to the membrane. अब इसमें हमें 3 स्टेप्स में चलना है: प्री हाइब्रिडाइजेशन, हाइब्रिडाइजेशन एंड देन वॉशिंग. प्री हाइब्रिडाइजेशन इज डन बाय 10% नॉन फैट मिल्क. ये नॉन फैट मिल्क solution is prepared in TBS buffer and we add this buffer or this non-fat milk in a glass dish, in a glass tray, and we place our nylon membrane in that glass tray and place it over the rocky platform or agitate it for 30 minutes at 37 degree centigrade. इस स्टेप से क्या होगा कि जो हमारे अनवांटेड पार्ट्स हैं membrane के, nylon membrane के, they are going to be blocked and then we can proceed further. Once this incubation period is done, then we need to decant and discard the milk solution and we need to do the washing. The TTBS is going to be used as a washing solution for 5 minutes at room temperature, and 2 times this washing step needs to be repeated so that the milk solution can totally be removed from the membrane. Now we are going to add minimum volume of antibody solution required so that it covers the membrane, and we are going to incubate the membrane in the presence of antibody solution on a rocky agitatory platform for 1 hour at 37 degrees centigrade. After this incubation period, we have to perform the washing, and that washing is done by TTBS for 5 minutes. Each washing is done and the 3 washings are done. So इस particular incubation period में क्या होगा कि जो हमारी primary antibody है that will bind to the antigen of interest, and then we have to identify this primary antibody using the secondary antibody or protein A peroxidase we can use in place of secondary antibody. The same thing is repeated. Add the secondary antibody solution or protein A peroxidase solution, incubate at 37 degrees centigrade for 1 hour. After that, remove this solution and do the washing for 4 times for 4 minutes each, and the washing will be done 3 times using TTBS. Now this secondary antibody is in place where the primary antibody was attached. Now we need to add the substrate. The substrate is going to be diaminobenzidine solution. Once we add it wherever our enzyme is present, the enzyme substrate reaction is going on, and then we can see that the color development starts. This color development will be completed within 5 to 10 minutes, and then after 5 to 10 minutes we can rinse this membrane with water. We can place it over the filter paper and let it dry, and then the signal can be recorded on an X-ray film using any available X-ray film and X-ray developing machine. We can see the black dots or black bands on the X-ray film that reflects the density quantity of the protein which is of specific antibody and antigen complex is the reason for appearance of that particular band.